Muy buenas tardes a todos. Vamos a continuar el día de hoy con nuestra serie de simposios que realizamos los días lunes y jueves, nuestros ya tradicionales simposios, pero hoy no es un simposio más. Hoy contamos con la presencia de la Fundación Oftalmológica Nacional de Bogotá, Colombia, en manos cuyo director es el doctor Francisco Rodríguez. Y entonces, en base a nuestra amistad de casi 30 años y a nuestra relación actual, hemos decidido realizar este simposio en forma conjunta. Entonces le voy a dar la palabra a mi amigo Francisco para que él introduzca el simposio, explique de qué se trata y explique quiénes son aquellos que van a participar del mismo. Francisco, es un honor poder contar con tu presencia y con la presencia de la Fundación Oftalmológica Nacional. Así que dejo el simposio en tus manos. Un gran saludo.
Arturo, buenas tardes. Muchas gracias por tu colaboración para poder realizar este simposio en conjunto con la SAO. Es un honor para nosotros compartir este momento y además un gran honor poder contar con estos personajes que han decidido acompañarnos para hacer sus presentaciones sobre un tema que yo creo que es muy relevante en este momento, que es la nutrición y la degeneración macular relacionada con la edad. Tenemos 3 expertos, más la coordinación de Patricio, que harán de este simposio inolvidable por las características que tiene. Agradezco a Patricio su gran ayuda en la coordinación y moderación de este simposio y le doy la palabra para que haga la presentación de estos 3 grandes invitados que tenemos, a Emily Xu, a Rufino Silva y a William Lee, quienes nos deleitarán con sus presentaciones. Patricio, muchas gracias.
Muchísimas gracias, Francisco. Muchísimas gracias, Arturo. Bueno, vamos siendo que es un grupo de speakers internacionales, vamos a hacer para poder la presentación de estos 3 speakers. So today we're going to have 3 speakers with us today. That's Dr. Emily Chu, Dr. Rufino Silva, and Dr. William Lee. These are 3 main topics that will revolve about nutrition and what is the effect on aging and our eyes and, of course, macular degeneration. So our first speaker is going to be Dr. Emily Chu. She is a graduate from the University of Toronto as an MD. Then she trained as an ophthalmologist at the University of Toronto. Then she did her medical retina fellowship at the Wilmer at the University of Nijmegen as well in the Netherlands. As her main positions, she is a former president of ARVO. I remember her walking around ARVO, being at every single of the talks there. And she's also a former president of the Macular Society. She's the National Eye Institute Deputy Clinical Director, but she's the head of the Division of Epidemiology and Clinical Applications, from which several of the main trials that we know these days have been completed. One of them, ETDRS, the diabetes study, the ACCORD study as well, the Action to Control Cardiovascular Risk in Diabetic Patients, and the ARID study. We had so much impact in our clinical practice. Also the ARID-2 these days, she is working on a MACTEL project as well. So Emily, the floor is all yours. Let me just bring down my screen so you can bring your slides in. It's a pleasure to have you here. There you go.
Okay, perfect. That's good. So I'm unmuted. Thank you so much. Thank you for having me. It's a great pleasure to be here. I'm going to speak about nutrition and macular degeneration. As you well know, we've been, and thank you for the lovely introduction. ARIDS is one of our main studies that we've worked on for actually a couple of decades now. So, and I have no financial disclosures for this presentation. So macular degeneration is the leading cause of blindness in the United States, and it's third in the world. It's predicted in 2040 that 288 million people will actually be affected by this. So this is a huge number we're talking about. There are a number of risk factors. I'm going to hone in on nutrition today. So just to be certain that we know we're talking about the same thing, I'm going to give you a definition of what this may be in terms of macular degeneration, what our definitions are. Early macular degeneration are those with medium drusen, intermediate AMD are those with a large drusen as seen here. And the late stages are neovascular, the wet form, or geographic atrophy, either centrally involved or non-central, as you see here and on the fundus autofluorescence. So those are the potentially vision-threatening aspects of AMD. We know that genetics account for 60% of disease. And there are many genes now identified, 52 SNPs and 34 loci. So this is very interesting. And I'll allude to this a little later. I'm not going to go into much about genetics, but I want to talk about genetic interaction with some of the diet work that we looked at. So our first clue that macular degeneration was related to diet comes from a very large study, the NHANES, which is really an old study, which has been sort of the forefront telling us that the diet is really important. People who ate rich diets and rich in vitamin A, and it was inversely associated with macular degeneration. And from there, we actually did a study called the ARIDS, as you heard earlier. So the ARIDS, let me just go back. The ARIDS is a clinical trial that has a clinical trials component from 1992 to 2001. And from 2001 to 2005, it was the follow-up study. And we had 57 participants who were enrolled in the study. And this was chaired by Dr. Brick Ferris, and this is our investigative group. There was 11 clinical sites in which we enroll these patients with no AMD and people with early, intermediate, and late AMD. So it was a variety of condition here. We tested the clinical trial, tested really a daily oral dose of vitamin C, vitamin E, beta carotene, and zinc. We did this because at the time there was a lot of studies looking at vitamins for cancer and for cardiovascular disease. And sadly, none of those studies actually were positive. This is the only study in which 25% reduction was seen in progression of late AMD over the period of 5 years. So this combination was the best treatment. So here we are in the duration of the clinical trial. You can see that antioxidant and zinc, the combination gave the best reduction. And this is what 25% reduction in 5 years. We followed these patients for 10 years, and we actually gave them, in fact, everybody was on antioxidants and zinc. So by the time you finished clinical trial, if you were in the placebo group, you were given the whole thing. And even after that, at 10 years, we found a 27% reduction after stopping the clinical trial. So clearly, this is an interesting finding. So who would recommend it to? Patients with intermediate AMD, those with bilateral large drusen, patients with late AMD in one eye, and not for current smokers. Beta carotene increased the risk of lung cancer in 2 really large trials that were NIH sponsor. One was done in the Finland patients who were smokers, and then there was one in the Americans. And those 2 studies showed that beta carotene increased the risk of lung cancer by as much as 28%, and 8% increase in mortality. So often people who have children of affected individuals often ask whether they should be on an ARID supplement. And we should say no, because it doesn't seem to in fact improve those cases in any way. And so we really think it's a secondary intervention. The public health significance is that it reduces about 3,000 people from getting late AMD over a period of 5 years. So this is the timeline. In 2006, we started ARIDS 2 because of the dietary information we got from ARIDS 1, showing that people who eat a lot of lutein zeaxanthin found in spinach, kale, and collard greens, and dietary omega-3s in fatty fish, that there was a reduction in macular degeneration. This is done looking at questionnaire. This is on the left is the ARIDS questionnaire, and the right is the ARIDS 2 questionnaire. And at baseline, if you have a very high intake of lutein zeaxanthin, you eat lots of spinach, for example, you would have a lower risk of neovascular AMD, geographic atrophy, and large drusen. And this is like 25 to 40% less AMD with higher intake of lutein zeaxanthin. This is also true for taking fish like twice a week was supposed to be high. And you can see there's a 50% reduction in developing non-neovascular AMD. So it makes sense because omega-3 is a major aspect of the membranes of the retina. Lutein zeaxanthin is a major component of the macular pigment. So biologically, we think it's important. So ARIDS 2 was started to look at the effect of testing by adding the ARIDS formulation, what we have already, but adding lutein and omega-3 to it. And by doing so, we were looking at patients who already have macular degeneration, not what we did in ARIDS 1 when we had the variety of patients with AMD. these are patients with at least intermediate AMD or late AMD in one eye.
Emily,
So in this
Emily,
case, yes.
sorry to interrupt. I think your slides, for some reason, are not moving forward.
Oh, really? They're moving forward
Yeah.
here. Oh, I'm sorry. I can see them here.
Shall we bring the your presentation down and bring it up again so
Yeah,
we
sure.
can make sure the...
Okay. I'll stop sharing. Okay. I'm so sorry. You should have interrupted earlier.
No, that's fine. That's fine.
Okay. When did it stop? Where did it stop? But which slide would you say? Now I'll start back up again.
I think it was on the third slide.
On the third slide? Oh my goodness. Okay. That's not good. All right. Let me go back. On the third slide. Okay. Okay. Let me see if we can go back to sharing again. Can you see it now?
Yep. And I think we went through slide number 5. That one was seen. Yep. Yeah, the SNPs. Yep, that was the introduction. That was seen as well. And the first that presents your results of the ARID study with the first of the curves over time of the risk. That's the one that got stuck.
Okay. All right. How about now? Can you see it now?
Yeah, I can see it moving forward now.
Okay. So this is now just talking about who we recommend for the ARID supplement. Can you see that? Okay, great.
Yeah, excellent. Moving.
Okay, so this is now the timeline showing that ARIDS 2 we were going to start in 2006, and these again the kale and the lutein, the high intake of lutein and omega-3. This is the frequency questionnaire that we use to capture everything. And as we mentioned, the highest intake at baseline was associated with a reduced risk of having macular degeneration, both with the lutein, zeaxanthine, and omega-3. And this is the macular pigment seen in the fovea of a rhesus monkey, a primate monkey. It's very much similar to our macular pigment, showing that this is important. Lutein, zeaxanthin make up this macular pigment. We cannot actually produce it without having a dietary intake. So we think this is very important. And omega-3s are important because the membranes make up the omega-3. So the ARIDS 2 was adding lutein, zeaxanthin, and omega-3 to the original ARIDS formulation. So we really are adding to what we know already. And these are patients who already have macular degeneration, either large drusen or late AMD in one eye. And so we had a secondary randomization looking at beta carotene because of the problem with the with lung cancer. And we also looked at reducing zinc. So this is the randomization. We had patients in all 4 groups, lutein, zeaxanthin, omega-3 in a combination. And then we asked the patients whether they would be willing to do a secondary randomization, which is looking at lutein versus beta carotene, for example. And these are the findings. The primary randomization showed that omega-3 actually had no effect, neither beneficial nor harmful effect. But the lutein zeaxanthin had an incremental increase. It was slightly better than beta carotene, especially in those cases in where people have a very low dietary intake. People who never eat spinach or collard greens or any of those things, they had a 25% reduction in having the lutein zeaxanthin. Whenever you directly compare beta carotene to lutein, there was always lutein always favored. It was always at least 20-25% improvement. But the thing that really clinched it was the fact that there was a 2-fold increased risk of lung cancer in those patients taking beta carotene versus lutein. And 90% of those were former smokers. So beta carotene, even though you stopped smoking, you still had a risk for developing lung cancer. So for safety reasons, we substituted beta carotene with lutein zeaxanthin. And so this is the ARIDS 2 formulation that we use now, which is vitamin C, E, no beta carotene, but lutein zeaxanthin, the zinc and copper, and no omega-3s. I want to say a word about the Mediterranean diet, because we have some interesting data that's about to come out pretty soon. It's just been accepted in the literature. And as you well know, Mediterranean diet is important for cardiovascular disease. This is first noted in the island of Crete, where patients were found to have low level of cardiovascular disease, and they thought perhaps it's because of the diet that they take. And there is, as you well know, there's an excellent trial looking at the Mediterranean diet for cardiovascular disease conducted in Spain. And this is looking at Mediterranean diet versus a controlled diet of low fat. And if this reduced cardiovascular disease by as much as 30%. So this is a good clinical trial evidence that Mediterranean diet has a role. And this question of Mediterranean diet with AMD has been looked at. Some of you have been involved in some of this, both Europeans and also the ARIDS data, the longitudinal data, the iRisk consortium from the Europeans looked at the longitudinal method, and then there's a cross-sectional way. And when we looked at these data, there was a 25 to 40% reduction. If you have a high adherence to diet, you're really very compliant and take a very high Mediterranean diet, your rate of AMD does reduce. Sorry. And this is, what is Mediterranean diet? It's low on meat, refined sugar, and high on other things like fish and whole grains, vegetables, nuts, and legumes. And again, we use dietary questionnaire, and we combined our two studies together, and we found that we had over almost 8,000 patients in the study, and a little bit more women than men, and they're slightly older patients in ARIDS too, because of the fact they all have macular degeneration, and they're not a great number of smokers, because these were clinical trials. People were probably much more health conscious. And we looked at the alternative Mediterranean diet, which is the American diet, which is not quite your adherent to European diet. So we had patients with no late AMD at baseline, and we had almost 8,000 patients. And we looked at the Mediterranean diet, looking at highest intake versus moderate intake. So you see by this forest plot that anything to the left of one is protective. So it's protective for late AMD and for geographic atrophy, as well as for neovascular AMD. And here are the numbers. You can see that these hazard ratios are within Bonferroni correction, because we're looking at so many aspects of this. We did a Bonferroni to correct for the p-value to make sure that we're not doing multiple testing and coming with something that's statistically significant, which is not true. So we see that clearly there is almost a 30% reduction, especially for geographic atrophy, with a high adherence to Mediterranean diet. And so this is for both neovascular and late and GA. And what drives it is actually fish. Of all these components, we looked at all different components, and fish has a highest intake of 0.69 hazard ratio. So this is almost a 31% reduction in the risk of progression to geographic atrophy. This is in ARIDS. When we look at ARIDS for large drusen, for the progression to large drusen, these are patients who didn't have large drusen. This is the early end of the stage of the disease. Again, Mediterranean diet is important. Even in the early part, you can see that the high intake reduces the risk by about 25% reduction in the rate of having large drusen. So even if it's if you're in an intermediate stage, even an early stage, there seems to be an effect. And these are the findings that we found. The combination of the two, and what really drives it is really the geographic atrophy. Most of it is in the geographic atrophy. That's particularly important because we don't have good treatment for geographic atrophy. And so this is summary for fish. Again, fish is actually very important. So I'm just going to skip through this. This really says that it's really important for the Mediterranean diet. It's never too late to start the Mediterranean diet. These associations, of course, are not results of clinical trials. I'm going to just say a few words about cognitive function. We're able to do that in ARIDS 2 and ARIDS 1. We know that Alzheimer's is really a significant problem worldwide, and we've looked at omega-3s as a randomized trial. We found nothing there. But the observational data is, again, very interesting. We looked at telephone ticks, the so-called mini-mental, which is sort of the key study for cognitive function. We did a variety of other testing done through the telephone, and this doesn't involve any visual function at all. We tested at baseline and every 2 years, and we found again that with cognitive function, and we just had this published in Alzheimer and Dementia Journal, that with tertile 3, the highest intake, the odds ratio was 0.36 and composite was 0.56. So there is a very good reduction in the risk of having cognitive impairment, again, with Mediterranean diet. A high adherence diet was important for your cognitive abilities. And when we looked again at the, and this is for AREDS2 is very similar. So these patients are older, but yet still the Mediterranean diet still had an effect both. And an interesting thing is there's like a dose response. The more you ate, the more the reduction there was. And this made it kind of interesting. And again, what pops up is fish as being important for cognitive function. Again, this is a very interesting hazard ratio, 0.36. This was, again, helpful, even statistically after Bonferroni correction. This was true in AREDS1 and not so much in AREDS2, because AREDS2, I think the patients are older individuals. And this is AREDS2. The fish, again, was important, again, at 0.54. So this is a pretty interesting, I think, data. If people can eat well, they can really help themselves, we believe. So, so the closer adherence diet to like a very highest intake, there's a lower risk of cognitive function. Again, component fish again pops up, and this is really important. Just a quick word about genetics. We know about complement factor H in 2005, and since then we've got tons and tons of data suggesting that there are many SNPs involved in 34 loci. And I just want to talk about the genetic interaction. If you have a diet and you have a genetic disposition, is there an interaction? Does it affect you if you have a certain genetic component of composition? It doesn't look like that for Mediterranean diet. We found absolutely no dietary recommendation, no genetic testing when there was no interaction with Mediterranean diet. However, with the fish, especially for geographic atrophy, we found that there was in particular for the cognitive function itself and in terms of complement factor H for Mediterranean diet, there wasn't anything there. I'm jumping the gun here, sorry. But for fish, there certainly was. And I'm not going to go through that much more, other than saying that there was a really important, with the protective allele, there was a reduction in having cognitive function. So the more protection you have, the better you should take the diet. So in summary, AREDS2 formulation is recommended for patients with intermediate AMD or late AMD in one eye. Genetic testing may be encouraged for research purposes, and it didn't really imply in our clinical care, at least for diet or for treatment with other things such as AREDS supplement. And the importance of Mediterranean diet is important for both AMD and cognitive function, and it's important for even early and intermediate AMD. So remember, we are what we eat. So I think it's very important. I'd like to thank the AREDS investigators for all their help and of course all the patients and all my collaborators at NIH, and thank you very much for your attention.
Thank you, Emily. That was a great talk. We're going to leave all the questions for the end of the talks, okay? So now we're going to have, let me just, can you bring down your talk now, Emily, so I can?
Yeah, I'm trying to do that now.
Okay.
Oops. Can you just, I can't seem to do that. Okay. Is that good there?
Excellent. Perfect. Thank you. Okay, so our next speaker is going to be Professor Rufino Silva. Rufino is a good friend. He's from Coimbra in Portugal. Coimbra is a very old city within Portugal. His education was all done at the University of Coimbra, where he got his MD degree and his MSc in ophthalmology, all from the University of Coimbra. And then he went on to have a PhD in ophthalmology. He's a professor of ophthalmology at this unit, and he's a great researcher. Many of the papers that we use these days, for example, and I'm quoting just one of them because there are hundreds of papers of his authorship, is, for example, the trend, that trial that evaluates the treat and extend versus the monthly injections. So Rufino, he's from this University of Coimbra, which is the oldest university within Portugal. So Rufino, all yours.
Thank you. Thank you, Patricia. I'm going to share my screen. I think now it's okay. So I want to thank the invitation to be here and to present this talk, the role of Mediterranean diet in AMD. Thank you, Francisco. Thank you, Arturo. Thank you, Patricia. And it is a great pleasure to be with Professor Emily Xu and William Lee. Thank you again. These my financial disclosure. They are not related with this presentation. And during the next minutes, I will try to answer this question. The first one, some of these aspects were already presented by Professor Emily Xu. So the first one is, what is the Mediterranean diet? The second question is, can the Mediterranean diet reduce the risk of early AMD? And I will present some results from our study, the Coimbra Eye Study. The third question is, can the Mediterranean diet prevent the progression to late AMD? So we have a beautiful paper from the Eye Research, the Eye Risk Consortium. And the fourth one is the effect of the Mediterranean diet affected by the genetic susceptibility? So we have information coming from the Rotterdam study and also from the RS study. So let's move to the first question. What is the Mediterranean diet? Professor Emily Xu already explained it very well, what is the Mediterranean diet. So it started in the 60s. Some people realize that the population from Crete and Greece, they have healthy aging and less cardiovascular diseases. So people living around the Mediterranean, it has at least 17 countries, but not, of course, not all of them have Mediterranean diet. It includes, for example, Spain, Greece, Crete, Italy, and also Portugal. Portugal has not the Mediterranean Sea on its borders, but it has a Mediterranean diet. So it's based on traditional foods that people use it to eat in these countries. And it showed to be people with a high adherence to this diet, showed to have lower rates of mortality, chronic diseases, stroke, as well as healthy aging. What do we have? We have not only the type of the kind of foods, but also daily physical activity included in this concept of diet. And we have something that now is forbidden in relation to because of the COVID-19, eating with family, big families, taking time to eat. So, and this is somehow that we use it to have now in Portugal. So it includes the daily physical activity, eating with family, but also so the daily servings, including legumes, nuts, bread, whole grains, including fruits and vegetables, olive oil for unsaturated fatty acids, fish and seafood, at least few servings per week, and less dairy products, cheese, less poultry, and even less red meats. So this is the concept of Mediterranean diet. And the important thing is that we have a score to evaluate how is the adherence to the Mediterranean diet. So the score is 1 or 0 in relation to the median sex-specific food group consumptions in grams. So it's easier to understand this, because if someone has an intake that is under the median for the sex-specific type, so for the good ones which are vegetables, fruits, legumes, cereals, fish, and the monounsaturated fatty acids, so it is under the median, the score is 0. If it is over or is above, is 1. For the bad ones, meat and dairy products, if it is under, the score is 1. If it is above, the score is 0. And the moderate consumption of alcohol is considered good. So the range is between 0 and 9. And this score is very, very useful for doing research. So this is the first question. The second question is, can the Mediterranean diet reduce the risk of early AMD? This was, Professor Emily Xu has already shown that, in fact, yes. I'm going to show the results of our study. We did a study in Coimbra. We call this the study of 2 cities. It's interesting because we analyzed 6,000 people in 2 different towns and all of the images were classified by a reading center. And an important thing is one town was an inland, and the other was a coastal town. And in fact, the prevalence in more or less 3,000 people in each town were studied, all of them 55 years or more. And the prevalence was almost double in the inland town. And this was somehow surprising, but the reading center was the same, all the methodology was the same, so this was quite surprising for us. Now, in both populations, we did a diet and lifestyle study. And for the LOSA study, so the inland town, we evaluated the protective role of the Mediterranean diet with the food frequency questionnaire, and also the impact of some nutrients, the dietary nutrients. In this population, we studied 800 people, 883, and half with AMD, half without AMD. We used the food processor plus to evaluate the nutrients. We used the quantified questionnaire, food frequency questionnaire, and the MEDI score. So what we found that those with the higher adherence to the Mediterranean diet, they had less AMD. We are talking about early, intermediate, more or less, because we have a few cases of late AMD, but everything is included. Most of the patients had early, intermediate AMD. So there was a reduction of the risk when the score was 6 or more. We did the same study in the coastal town, and we found also the relation with the higher score for the adherence to the Mediterranean diet, and then we analyzed it together, the data together, including more than 1/3 with AMD and 2/3, near 2/3 without AMD. And we found that, in fact, the population with the lower prevalence of the disease, they had a higher adherence to the Mediterranean diet, and the difference was significant. So definitely there is a role for the Mediterranean diet on the prevalence of the early and intermediate AMD, and so the higher adherence to the Mediterranean diet is significantly associated with no AMD when we analyze these two populations together. And this no AMD was driven by the higher adherence to the Mediterranean diet, driven an increase in consumption of vegetables, fruits, and nuts. Surprisingly for us, in this population, fish was not one of the foods implicated in this reduction. The nutrients analysis were those that we know very well: vitamin A and C, and carotene, and iron and zinc. Also the monounsaturated fatty acid. We also a role for caffeine, which as a statistic find it, we don't know, we need to have other results. And regular physical activity alone was also associated. So this is somehow that is important. It's a concept, not only the type of foods. So the answer is that the high adherence to the Mediterranean diet is associated with a lower prevalence of early intermediate AMD. So this is the second question. So now we are going to move to the third question. The third question is, can the Mediterranean diet prevent the progression to late AMD? So this is an interesting study from the IRIS consortium. And the objective of this work was to investigate the association between the adherence to the Mediterranean diet type and the incidence of advanced AMD in a large population prospective sample. The IRIS includes 17 European studies. So this is a huge consortium. We are also part of one of the studies. 8 of them were prospective, but for this particular aspect of evaluating the importance of diet, we only have data from 2 studies: the Rotterdam study, so the incidence of late AMD, 117, and 38 from the Alienor study. This is in Netherlands, this is in France, and this was after 9.9 years for the Rotterdam study, and after 4 years for the Alienor study. But the important thing here is that they used the MEDI score. It was, you know what it is already, and they found that near half of the patients had a median score, and near 1/5 were in the high score. And this was similar in the Netherlands and in France, as you can see here. So the interesting thing is that those patients, the population with the higher adherence to the Mediterranean diet, had a significantly less progression to late AMD, as you can see here. And this was replicated in the 2 studies. It's interesting. So high adherence, less progression to late AMD. And when both populations were analyzed together, the difference was significant. And the effect was driven by fruits, legumes, and fish. So we are talking about the progression to late AMD. So the pooling data of this the study showed that the higher adherence to the Mediterranean diet was associated with at least 41% reduced risk of incident advanced AMD. So this is the 3rd question. Now the fourth question is the effect of the Mediterranean diet affected by genetic susceptibility? We have data coming from the Rotterdam study from 2011. And this is an interesting thing. This is like having a good diet means that you can eat the genetic risk, something like that. So you have, this is a CFH genotype. As you can see, the green ones, if the patients, if the population has a higher intake of zinc in the 3rd tertile, so the risk decreases. The risk is similar to be non-carrier or be heterozygous. So the homozygous, if they have a good diet with zinc or with beta-carotene, or even with EPA or lutein, there is a decrease in the risk. The same from here, the logs 387715, the genotype, the non-carriers, the risk becomes similar to be a non-carrier if there is a higher intake of zinc or EPA. So it's something like the antioxidant intake diet decrease the risk of early AMD in those at high genetic risk. This was presented in 2011. We need more information. But the higher adherence to the Mediterranean diet was associated in this case with a decreased risk of 26% of progression to late AMD. However, among the subjects with high genetic score, risk score, the protective effect was there, but the difference was not significant. So it's not the same for everyone. And remember, we are talking about genetic risk, and we are including also, and when we talk about intermediate AMD, we are not talking about a single entity. This is a wide range of severity, so probably this needs further study. And Professor Emilio Schul has shown, for example, the progression of drusen, which is important. And in this case, also the MEDI score was not associated with AMD among subjects who were homozygous for the risk of LCC. So in these cases, there was no difference. The Mediterranean diet has no effect. So to conclude, what is the Mediterranean diet? As you have seen, is a healthy and easily accessible diet, originally made by people living around the Mediterranean Sea with a high consumption of vegetables, fruits, legumes, cereals, fish, monounsaturated fatty acids, and low consumption of meat and dairy products, and a moderate consumption of alcohol, and includes also exercise and have a meal with time and with the family. Can the Mediterranean diet reduce the risk of early intermediate AMD? Yes, we have also our study and also other studies. It reduces the risk of early intermediate AMD, and the food group analysis showed that it's a significant role for vegetables, fruits and nuts, legumes and fish. And the nutrients are those we know very well. Can the methylphenidate prevent the progression to late AMD? In fact, yes, at least between 26 and 41%. And is the effect of methylphenidate affected by the genetic susceptibility? Apparently, yes, and the effect seems to be less expressive when there is a high genetic risk score. So this is what I had to present. And now I give the word to Patricia. Thank you.
Thank you, Rufino. Thank you very much. That was a great talk. Great talk. I'm just getting starving here, you know, talking about fish, and I'm getting some of the questions ready. So I'm looking forward for the end of the session. So let me just share my screen again so I can introduce the third presenter of the session.
So I need the... Okay.
Okay, and the third, last but not least talk is by William Lee. Dr. William Lee, he's a good friend. His education was done at Harvard College, then he got his MD degree from the University of Pittsburgh. He did internal medicine at Mass General, the University of Harvard. And his position, he is the CEO, president, and co-founder of the Angiogenesis Foundation. The Angiogenesis Foundation is a well-known organization that helps people get together with ideas and tries to find gaps in areas where research is scarce and needs to move forward. But he's a great speaker. You can see many of his TED Talks, or when he goes to the famous TV shows in the US like Dr. Oz or many others. And he wrote a book recently, which is called, according to the New York Times, this is a bestseller. It's a book that it's called Eat to Beat Disease. So he's going to be talking about these, but also he's got a bonus for us at the end of the talk regarding some research he is doing related to coronavirus. So I will leave the space for you, Will, to go ahead with your talk.
Great. Thank you very much, Patricio. I would like to thank Dr. Alessandrini, Dr. Rodriguez, Francisco, a good friend of mine for many, many years, and nice to see colleagues like Emily, and very nice to meet Dr. Silva. A real pleasure to speak with this group. I speak frequently about nutrition and the effects on the body from the perspective of having been involved with drug development and understanding that what we put into our body, whether it's a pill, an injection, or food, activates many similar pathways in the body that can have an impact on physiology or pathology. And so I wanted to share with you a couple of insights onto this in the era of COVID-19, which affects not only our patients, of course, but also affects us as providers when we are actually exposed to patients, or also when we're trying to make some advisement to our families as physicians. And so I'm going to share with you some unpublished data talking about COVID, but I think it's so important that we openly share information that I didn't want to actually wait for a publication. So let's see, I am trying to advance my slide here. Okay, so this is actually a very, very important article just from last week from the journal Science that really articulates how the coronavirus enters the body and what we know so far about its impact into the system. And I want to make the case that surprisingly, the coronavirus is an angiogenic disease. It has a microvascular component, and that there's a very real possibility, although conjunctivitis is typically the most common observation to date, that as this pandemic continues and we have the ability to look deeper into our patients, potentially autopsy tissue as well, we may find some interesting correlates in the retina, and I'll try to make that case. So we all know that COVID-19 is a respiratory virus, a RNA virus that invades the lungs through the mucous membranes. We know that if you actually have high titers of protective antibodies, you should in theory be more protected, and that the virus can invade the ACE2 receptor producing cells in the upper airway epithelium, but and that's probably where the first battle occurs. But later, if we haven't been able to overcome that it then descends into the lungs, where it actually has some potentially devastating consequences. And so the tragic scenes that we all appreciate as healthcare providers happening in our hospitals really are the people who have been succumbed to a greater viral load that actually has compromised our airways in a substantial way. So what's actually happening? Here's actually what I want to share with you. On the upper left-hand panel is an autopsy specimen from a patient who died of COVID-19. This is a European patient, and you can kind of see, although the lung looks edematous and hyperemic, what you're actually seeing on the upper right-hand panel, in fact, are dilated alveolar ducts thickened with inflammatory and T-cell infiltrates. That's on the upper panel up here. If you look below, you can see that within the capillaries of the alveoli, it's filled with microthrombosis, not so much in the large airways or large vessels leading to the airways, but actually in the small alveolar vessels. And on the lower right-hand side, you can see by immunohistochemical staining, high populations of CD4 T lymphocytes that have gathered around as well. Now, what I can't talk too much about because of time, and also the data is still being analyzed, is looking at the relative absence of B cells, antigen presenting cells. Also, we found a 25-fold increase in ACE2 presenting alveolar airway cells as well. And so this is actually starting to look at the ultrastructural level, some of the things that we initially thought were beginning to do a deep dive into the tissue. Now, why am I telling a group that's interested in ophthalmology is that one of the things that I think is very important is for us to be able to cross-fertilize knowledge about the microcirculation and about pathogens and disease pathology from one area of the body to the other to see what we can actually learn. So now let's go deeper down in the upper left-hand column here. This is actually a scanning electron micrograph with corrosion casting on the far right of a COVID-19 patient who has actually succumbed from their disease. This is a patient who died of respiratory failure. And you can see there is aberrant, chaotic microcirculation. The actual blood vessels from this corrosion cast of the lung from the patient affected with COVID is very unusual and different compared to the normal lung microcirculation. So this is really a very, very interesting morphological change you can see between the infected lung versus the normal lung by scanning electron microscopy. And if you look more closely at the area which is on the upper left-hand panel on the lower left-hand white box, and you take a look at the deep dive, you can see areas of attempting to sprout angiogenesis from rapid hypoxia occurring probably from this microthrombosis. But you can also see in these red arrows, areas of intussusceptive angiogenesis, which is an alternative pathway for angiogenesis in which there are pillars of connective tissue descending from the roof of the blood vessel and ascending from the bottom of the vessel. It's sort of like the stalactites and stalagmites forming to split a vessel in an effort to actually get better circulation. Now, obviously, these are not well understood exactly in COVID-19, but I wanted to share with you some microcirculatory changes that are occurring in a hypoxic and potentially getting to an ischemic state. Now, the heart also has, I'm not going to go into the heart, but also has microcirculatory impact where we know that the ACE2 receptors are on the endothelial cell surfaces within the myocardium. People are presenting to the emergency room with chest pain, brought to the cardiac catheterization lab, and found that they have no occlusions in their coronary arteries, and yet they actually have some form, it was called myocarditis, but we believe it's a cardiac microangiopathy. Again, still, we're working on this right now to look at what's happening. And in the brain, which is closest to the eye, central nervous system, we're also seeing some patients actually having CNS signs, whether it's a stroke or seizure, confusion, thinking some brain inflammation. And what's very interesting is that there is actual encephalitis that's also seen in some patients infected by COVID-19. And so that leads to the thought that perhaps this virus does actually penetrate through the blood-brain and blood-retina barrier potentially, and that in the future, as we begin to look at this disease state, we might be thinking about what some of the microcirculatory changes would be, particularly in an elderly population that may also have forms of either dry AMD or wet AMD, early or intermediate or late stage. And so I'm setting this out really to share with you some really hot out of the press, or not even the press, hot out of the oven data looking at the microcirculation, because this is something that the retina specialists really have a great interest in. Now, because we're talking about diet, I want to share with you some of the approaches that are now being considered in dietary interventions of COVID-19 for either prevention, treatment, or recovery. And this is really the area of molecular nutrition, looking at impacting diet on angiogenesis, diet and immunity or microbiome, diet and regenerative changes, or potentially in antioxidant components of DNA. This is just an early area that's starting to be written about, mostly in the looking at the micronutrients that can support healthy immunity from associations and population studies. These are some of the micronutrients that are found in foods that you can find in the grocery store or the market very easily. Some of these vitamins, you can see the zinc and vitamin C are common to the, for immunity, common to the previous talks that were given by Dr. Chu and Dr. Silva. But I want to actually introduce another deeper layer that's being looked at outside of COVID-19, which is dietary bioactives beyond the classic micronutrients. And these are some of the same chemicals that give foods their colors, their flavors, and potentially attract insects for pollination. They can be upregulated by injury to the plant. They can be, they're seasonal changes in terms of the concentrations. I don't have time to go into everything, but for the purposes of this talk, I wanted to share with you something that matters, which is the connection with immunity. I'm going to show you one slide about angiogenesis. That's a bigger discussion, but right now, what we're beginning to ask the question are there ways that both clinicians, ophthalmologists, and patients and their families, our families, can we actually do anything to support at the front line gate of defense through immunity, and what's the connection with our gut? The microbiome is a very intriguing area to look at, the 39 trillion bacteria compared to the 37 trillion or so human cells, a huge number of diseases associated with dysfunctions or aberrations in the microbiome, dysbiosis they call it. And some of the foods that have actually been associated with intake in small studies, this is just 6 female volunteers in Singapore who actually had their stool measured at baseline and then given kiwi, which is high in vitamin C, for 2 fruits a day for 4 days, and then checking changes in their fecal biome, and finding that, for example, some species of lactobacillus can change as quickly as 24 hours. And bifidobacteria, which is another helpful bacteria that can improve immunity and lower inflammation, increasing by 17% in terms of the beneficial short chain fatty acids. So this is another dimension that I think that we can begin thinking about conducting studies in vision and diet and looking at that connection between what we intake and what is the stool showing in terms of not only bacteria patterns, but also short chain fatty acids. We also know the microbiome is affected by fermented foods, whether you're looking at sauerkraut, kimchi, various pickles, or different Asian cultures have their own form of pickling. Many times these pickling processes allow bacteria from the air to settle on, they colonize, they overgrow, and there's a dominant species. And the ones that happen to be in these foods tend to be generally probiotic, meaning introducing healthy bacteria, which we're beginning to realize impact healthy immunity and also other metabolic states. So this is just a partial list of foods that affect the microbiome. I'm going to show you a slide that you can get all these lists on a little bit later. That's an area I'm working on. But what's the microbiome connection to the immune system more directly? I think this is a very interesting area for all of us who are thinking about our own personal health. And I want to bring the US former President Jimmy Carter, who is in his 90s. He's the oldest living president from the United States. He was diagnosed with malignant melanoma from his arm that spread to his liver and his brain. And he was one of the early beneficiaries of immunotherapy for cancer using a checkpoint inhibitor. So this is actually a drug called pembrolizumab that literally rips the camouflage off of cancer cells, revealing cancer to be not self, allowing our own immune system. In his case, even at the age of 90, a 90-year-old person's immune system is capable of spotting foreign bodies and foreign cancer cells and eliminating them. And he actually is now cancer-free. This is pre-treatment and post-treatment. This is not Jimmy Carter, but this is a similar patient with a response to melanoma. You can see very dramatic clearance of the disease. And what's interesting is not everybody responds to cancer immunotherapy. And in fact, one of the things that we're beginning to discover is that the gut microbiome, the presence of particular bacteria, in this case, Akkermansia mucinophila, which grows in the gut mucus and is influenced by intake of dark red fruits like pomegranate and cranberry, actually seems to be correlated with response in cancer patients to immunotherapy, meaning if you have this bacteria in your gut microbiome, you are likely to respond. This was published by Laurence Zitvogel out of the Institut Gustave Roussy in Paris. And if you lack it, you tend not to respond. And this has now been replicated over and over again. And what's interesting is what the mechanism of action is. So this is what we all want to know. It turns out that some of the elagitannins in pomegranate, for example, causes the gut epithelium to secrete more mucus, healthy mucus, that the Akkermansia normally thrives in. With higher levels of Akkermansia in higher levels of mucus, that bacteria interacts with the gastrointestinal lymphoid tissue to release all these other immune cells that are part of our immune system. When you actually have immunotherapy, which then impacts on these cells and is requisite to impact these cells, you can have an effect. Now, why do I show you this in a very personal way is because that bag of immunotherapy drug was given to my mother, who in her 80s was diagnosed with metastatic recurring endometrial cancer and given like zero chance to survive. And we got her on, we found the markers that would make her a candidate for immunotherapy. And we actually manipulated her diet to be able to actually increase her mucus and increase the Akkermansia population as she was getting this immunotherapy. And after 9 weeks of therapy, she actually had a complete response. And to this day is actually healthy. And in fact, living with me during the COVID-19 pandemic. And so this is one example that is now being replicated and studied in clinical trials and how to use diet. Here's another one that's more directly associated with virus. It turns out that broccoli sprouts, which contain sulforaphanes, actually seem to actually impact the body's response to vaccines against influenza virus. And so this was a study in North Carolina that was done with 29 healthy volunteers giving nasal influenza A vaccine. So this is putting partially killed vaccine into the nose of young, healthy people, and then giving half of them either 2 cups of broccoli sprouts into a shake to drink for 4 days, getting the vaccine. And what's really interesting is that within the broccoli sprouts, there's the sulforaphane levels of a broccoli sprout is 100 times the concentration of the adult big broccoli. And so they then delivered the nasal vaccine and they measured 4 days later the natural killer cells. Sprout eaters with the vaccine had 22 times more NK T cells. The NK cells had more virus killing power. And the sprout eaters also had fewer viruses actually in the nasal cavity as well. So eventually, another project I'm working on now is as we develop COVID-19 vaccine, I'm actually starting to set up some experiments to study whether or not the impact of diet can help to optimize immunotherapeutic responses to ultimately COVID-19. So these are some of the foods that have been studied in small clinical studies that influence immunity to virus infection, Again, I'm focusing on virus because of the connection to COVID-19, but they all have other bioactives that may impact on angiogenesis. And here's just some of the list of dietary components of foods that actually have various dietary bioactives that have been studied. And for those of you who want to get all the lists that I've shown, this is the book that I wrote, Eat to Beat Disease. All the lists, there's about 207 foods that I put in the tables, you can actually find there. And with that, I'd like to really thank the organizers for the opportunity to share not only some of the insights about diet and health and immunity, but also to bring you some of the new data on COVID-19. Thank you.
Thank you, Will. Thanks very much. That was a great talk, as usual. Let me just go back to the original presentation so we can just go into the questions. And I'm going to break the ice with some of the questions. The first one goes to Emily. Emily, you mentioned that the difference between ARIDS 1 and ARIDS 2 is that you tested during ARIDS 2 the effect of fatty acids, omega-3. And you mentioned that the results came up that there's no difference whether you include the omega-3 or you don't include it, there's no harm nor benefit. Although there's a European study that is called the NAT2 study, and they looked at it and they've done a further analysis looking at the red blood cells of these patients, and they found that there was a benefit in those that were properly receiving high doses of omega-3. What's your view on that, please?
Well, we studied that paper quite carefully. It's not a randomized trial they're looking at. They're only looking at patients based on their red cell level of omega-3. So it's not the actual randomized trial. So if you had a higher level of it, you had a more protection. So it wasn't necessarily that the supplement actually, because the actual study was negative in itself. So if you look at that small graph, it's really about that. We don't know what that means. Obviously, eating fish is important. So why doesn't omega-3 work? And it didn't work for cognition. And many of the studies that have looked at omega-3 supplementation has not shown to be important. So there may be something else that's important in the fish or whatever we're eating that's really very important. And we clearly don't have that answer.
Okay, so in summary, you wouldn't give to any patient at risk of, you wouldn't add the omega-3. Excellent.
I would not, no.
Rufino, you mentioned fish as well as in several parts of your study being one of the drivers for the benefit. Was it any fish or needs to be salmon? Salmon, is it the same farmed or is it the wild salmon that is better at, to be included in the diet?
So these are an interesting question, In fact, we had less AMD in the population in the coastal town. But when we analyzed the data, fish was not one of the drivers of the effect. For us, it was a surprise. I can't answer if the wild type of salmon and sardines, all of them, they are associated, or if I can answer it if there is any difference. I agree that fish is one of the drivers, for sure, because there's many other studies have that results. It's probably a question of statistics. I don't know. In our study, it was not one of the drivers of the effect.
Okay, but it wasn't the consortium, right? The consortium paper, the one that you mentioned, you showed at the end a meta-analysis of 2 trials, your trial and another study.
Fish was, yes.
So the overall response was fish was a driver.
Fish was the driver, yes. In the group, yes. But strange. It was strange that in the coastal town it was not one of the drivers. But they had also better diet in terms of having also other component of the Mediterranean diet. So it was not possible to demonstrate in this particular population.
Emily, is it any specific fish that you have identified when you looked at fish, or fish means any fish?
So we did not ask specifically for any fish, but if you look at some of the ingredients in each of the fish, what they're made of from our government has the USDA has actually talks about all individual constituents part of it. They do vary. So perhaps some fish are better than others, but we don't have that data either. It's just like Rufino, we don't really know which it is. We really didn't ask. I mean, it's always thought that the cold water fish are better, like even eating tuna and things like that. Even a can of tuna might be helpful, but we really don't have that answer. I don't have that answer.
Excellent. Well, Will, would you like to comment? I mean, is there any study that you have that you know of that has looked at this?
Yeah, well, you know, it's very difficult to tease apart all the different elements when you're talking about whole foods. And that's one of the challenges of nutrition research, and is correlations and also the complexity of the food itself. I will say that, you know, it is true, mostly it's cold water fish that have shown to benefit, but if you were to look more broadly at what's in seafood, because I think that fish is what we tend to simplify things to. Some studies have actually shown benefits across different chronic diseases for shellfish, as well as other open water oily fish. And I happen to be somebody who's done research on looking at other biological mediators of fish. And for example, in elasmobranchs, which are the kind of the whole category of sharks and rays, there are molecules called meganins and other small steroid-like molecules that can influence vasculature and other things that we don't haven't fully defined yet. And even in shellfish, for example, I didn't have time to show you some of the really interesting things in shellfish. There are proteins and polysaccharides that really influence immunity. They influence endothelial cells as well. And some of these studies have been done literally after having from the actual meat and extracts from commercial shellfish. So I think we're at the beginning of a new frontier to begin asking those questions, but we're not there yet.
Okay, thank you. And one of the questions coming from the chat is for you, Rufino. The question is, what if somebody with a high genetic risk, he doesn't follow strictly the Mediterranean diet? Will that increase his risk for developing the late disease?
Well, this is an interesting question, and I think many people is now studying and making a correlation with genetics and diet. We are also going deeply in this because now we have the genotype of our population, and we are correlating. First thing, I think we need to divide intermediate AMD. Emily Shu presented an interesting slide, drusen. For example, if you have drusen pigmentary changes, or if you have pseudoreticular drusen, probably you have a higher risk. The genetic risk is one part. And we know that there's 52 genes involved that probably have more, and we have even more rare variants that have a higher impact in the progression of the disease, and we know some of them we don't know yet. So these studies, remember, they checked only 10 genes, I think. They were addressed to the CFH, mostly of them, but now we have more genes to analyze. It's the idea I have is there is a lot to know yet. It is in the air the idea that those patients with a higher risk, or probably with specific variants or rare variants, probably the impact is not so evident. Another thing we should also analyze, and it's not analyzed in these studies, even in the population studies, is how is the intestinal absorption. So these studies were done in elderly people, so the age is over 55, but most of them with AMD, they have 70s, 80s. So probably Dr. Willie could comment this. So the intestinal absorption is not the same we are expected. I don't know how important is the serum levels of the antioxidants or whatever. So it's not an easy, it's an interesting question, but the answer is not only a unique question.
Okay, and there is a limitation with all these studies that look at the past of the patients, looking at the diets that they have had. And we sometimes think that there could be many confounders. I mean, maybe just living in the coastal areas of the Mediterranean make you happy, less stressed, and that's the whole explanation for, you know, having a better life expectancy or lower risk for developing wet AMD. But the ARED study didn't have that because it's a randomized study looking into the future of the patients. But maybe the confounder in that study was that many of these patients were American. They were having a complete, very good diet, and many of them were taking other vitamin supplements. Is that right, Emily?
Well, we control for the other vitamins because we actually gave them Centrum. So we didn't allow them to take other vitamins. So about 2/3 of the AREDS patient took Centrum plus the AREDS supplement. When we did the analysis with and without the Centrum, it made no difference. And then in AREDS 2, we gave them Centrum again, and that was really like 90% of them took it. But I think just to say a word about the genetics, I didn't present it very well in my presentation. I apologize. We did look at complement factor H and a genetic risk score and the ARMS 2 in the presentation that I talked about. What we did find was that there was an interesting interaction with complement factor H. It was the protective allele. So if you had the protective allele, you had a more, like you were already protected, but you had even a better chance of getting even less AMD. So it's like you can't afford not to eat it if you really had the protective allele. So that was kind of odd. But I think Rufino was right because it's just not genetics. There's many other things that might be the confounders that makes it very difficult. But clearly the Rotterdam study really is interesting. It does show like, you know, you can eat away your genetic risk. That sounds really quite interesting. But we weren't able to absolutely replicate everything. We didn't have the same type of population. And our diet probably isn't anywhere near as good as what Rufino looked at in your area because our Mediterranean diet just doesn't eat enough olive oil, for example. We just eat a little slightly different, we so-called modified Mediterranean diet. That may be one of the problems.
Excellent, thank you. And my last question, just to close the session, and thank you all the speakers for great talks, goes to Will. Will, sometimes here in Latin America, and you've been here, we went for some omakase sushi, and then we went for some grilled pork. But you know that sometimes it's not easy for us to access the best types of food. You know, some patients may have access to what they call so-called organic food. Do you think this is something we should push more the patients to go for this type of diet, you know, to make an economic effort and not eat the regular vegetables and fruits and go for type of, you know, organic food?
You know, I think that it's very difficult to get our arms around the impact of organic. It's conceptual. There's a huge amount of ideology about, you know, eating plants grown without the use of pesticides in a more naturalistic setting. And organic has traditionally been more expensive. Although I heard that in the United States, some of the big companies have actually committed to growing millions of acres of organic food. So potentially in the future, the prices may come down. But I think equity and access to food is most important to have to really just try to eat whole foods, mostly plant-based. Fruits and vegetables and legumes and nuts are very important. I will tell you an interesting thing that I discovered recently in talking to some plant botanists is that, and this was published in Nature as well recently, that in plants grown organically, the one thing that pesticides prevent is insects that would come to eat the plant. So not just the fruit, but they eat the leaves, they eat the stems. Those are considered wounds on the plant. And the wound healing response of certain plants like strawberries, where this has been studied to look at ellagic acid, is to increase gene expression of, for example, ellagic acid, which is one of the bioactives in the fruit. So the organic plant actually seems to have greater amounts of some of these bioactives, which may be one of the benefits of eating the fruits themselves. But comparative effectiveness of looking at these fruits has not really been adequately done yet, but it's an area of high interest to me. And I think people are asking this question all the time. I would say, if you can afford it, do it. But in any event, you just really should make sure that you eat more fruits and vegetables, much along the lines of what Rufino and Emily actually presented.
Excellent. I would like to thank all the speakers. We have a clear message here, which is we are what we eat, and we have supporting evidence for it. So I would like to thank you all. These were great talks. Sorry for extending the time a little bit more. We wanted to make sure we have the bonus of the COVID and all the questions answered for the people attending the session. So thank you very much. Time for dinner here, so we're going to try to eat the best we can.
Thank you.
Thank you.
Thank you.
for having
Thank
us.
you very much.
Bye-bye.
Thank you. Bye-bye.
Thank you.
Thank you, all of you. Have a good night.
You too.
Bueno, muchísimas gracias Francisco, muchísimas gracias Patricio, fue un excelente simposio, la verdad que tenemos mucho por aprender. Quiero agradecerles a los dos por la posibilidad de poder compartir con ustedes este simposio y bueno, nos estamos viendo a lo largo de esta pandemia. Les mando un gran abrazo a todos.
Un abrazo a todos
Un
también.
abrazo
Feliz noche.
también.